All right, ladies and gentlemen, welcome back. So, we are going to talk today a little more specifically about applications of the technology that we've been talking about thus far. So, today we're going to talk about GMOs or genetically modified organisms. So, let's just start off with a couple of quick terms just so we know what we're talking about here. So when we talk about genetically modified organisms, by definition, that's any organism that has acquired one or more genes by some sort of artificial means. So really, we're talking about utilizing that genetic engineering techniques that we were talking about with the recombinant DNA and the plasmids, or using some sort of virus, something like that, in order to get a particular trait that wouldn't naturally be found in that organism and giving it to them. You may also come across this term transgenic organism, where we're talking about a genetically modified organism who has received a new genetic material from a different species. Realistically, you may come across either one of these terms, and more or less, they mean essentially the same thing. So they're getting genes from somewhere. Um, I would say overarchingly that term genetically modified organism or GMO is probably more commonly used. But I wanted to throw both terms out there just in case you came across them. So if you look at the figure down here at the bottom, here we're creating a transgenic or genetically modified organism by taking a plasmid out of a particular agrobacterium. So it's a particular bacteria species that is really well known for causing infections in plants. And we're basically going to genetically modify it to not insert its sort of disease causing genetics, but instead taking a plasmid and creating recombinant DNA. And so we'll use those restriction enzymes to cut open that plasmid. Also use that same restriction enzyme to take a gene from some other organism that we want to insert. Let's just say it's one that confers or gives this organism a particular ability to have some sort of drought tolerance, right? So here's my drought tolerant gene, here's my plasmid that I've taken from this bacterial cell, and I'll use those restriction sites and use that recombinant DNA, create that recombinant plasmid. I'll now put that back into that agrobacterium and allow that bacteria to now infect that plant cell. When that does that, that new gene, that drought resistant gene, becomes incorporated in part of that plant cell's DNA. I plant the plant, and now this plant's going to grow up with that gene in place, and so it will be more drought tolerant. So it's allowing us to circumvent the need to do all sorts of crossbreeding and selective breeding, and hoping after many, many generations that I now have a drought tolerant plant. I can do this in a single step as opposed to sort of hoping and praying that my selective breeding processes are going to get me what I want eventually. This is a much quicker, more efficient way to do it, and more than likely, if I've done my genetic process correctly, I should be able to do it within a couple of tries or a few plants and have what I'm looking for. So, GMOs are overwhelmingly being utilized in our crops today. In fact, just a kind of quick statistic, I guess, if you look, over half of the US soybean and corn crops, at least as of when I put this PowerPoint together, were overwhelmingly genetically modified in some way, shape, or form. A lot of them are genetically modified for things like the drought tolerance, as well as things like pesticide resistance. So, if you know anybody who does any sort of farming, or you've ever come across things like Roundup Ready corn, they are corn plants that I can use a pesticide or a herbicide, excuse me, Roundup on, so that the farmers can go through, round up their crops, use this product, this Roundup on their crops, kills the weeds off so that the corn can grow and have more nutrients and be healthy in that respect. But we don't have to worry about the Roundup also killing the corn at the same time. So it's something that is very commonly used now in particularly stock crops for things like feed for animals and stuff like that. Other uses for it, we can use it to genetically modify organisms to delay their ripening, right? So if I'm getting bananas from South America, I don't want them to necessarily ripen as fast as they might naturally do because I want them to get to my grocery store shelves in Pennsylvania or New York or Maine or Canada before they go ripening. So that's a pretty common one that's out there as well. Improving nutrients. Um, if you've ever come across the golden rice, this is a pretty common one that was being utilized in third world countries. So, vitamin A deficiency is a big problem in those third world countries. It's not a nutrient that they typically get in their food, and as a result, you need that vitamin A for your eye production. And so, as a result, the people who created this golden rice, all right, it literally has this sort of gold character. I think a lot of vitamin A comes from things like your carrots and things, so it gives it sort of that orangey yellow color. This golden rice has extra above and beyond normal nutrients, specifically the vitamin A, and it's really helpful in those third world countries to give them a rice crop that is often typically a staple of their diet anyway, but give them this particular variation of rice to help with the eye production so that they're not going blind, particularly in children. We'll use it to do things like increase the size of the product, so bigger tomatoes, bigger strawberries, increase the yield of the plant overall, so just that you get more fruit from a single plant. Things like increased risk to spoilage and disease, the herbicide resistance, so there's the example of the Roundup Ready. We can even use it in certain situations to provide human vaccinations against certain organisms. An interesting one is things like the bananas that are used to help vaccinate against dysentery. So, dysentery is another sort of big disease that tends to plague third world countries, and we do have a vaccination against it. But in order to typically get a vaccine, you need a lot of things in place from medical staff to be able to administer it. Plus, a lot of times they need to be kept cold or something like that in order for them to be stable and not degrade over time. So, shipping becomes a problem. And you're talking about going into deep jungle territories or something like that, say in like Central America. Well, if we can ship a bunch of bananas in that have the vaccination or the vaccine as part of its process, and now you just have to eat a banana, the issues of the medical staff, the keeping it cold till it can get there, all that kind of stuff sort of disappears. And so being able to make those work is an interesting byproduct of some of these GMOs. Personally, I'd much rather eat a banana than have to get a shot. So if we can figure that out, I'm sure that would be something that lots of people would be interested in if we can make it work for more things. This is just a particular example that has been used thus far. We can also use GMOs in animals, and once again, this is often done a lot to our livestock, right? So beef, cattle, pigs, chickens, that kind of thing. Um, One of the things, obviously, is to just get a bigger animal, leaner meats. Maybe if we're talking sheep and wool for certain products, that could be a thing. Um, but we also do use GMOs for things like producing vaccines and growth hormones. Um, that growth hormone produced by bacteria can then be used as a supplement to increase milk production, growth hormones to just increase the overall weight and size of the animals. Um, we can even do some genetic modification to those animals to get them to produce human proteins that maybe we can't get a bacteria to do for us. So, all sorts of interesting things that we can do with animals as well. Now, that being said, there is definitely some controversy over using GMOs as part of our food staples. If you really start paying attention to labeling of things on the grocery shelves, um, there's a big question of whether or not GMOs should be used. Should they be labeled? Should we have government regulation? And the answer really is a lot of that stuff is coming um, and trying to be addressed. So there's definitely a lot more labeling than there used to be. There's definitely more government oversight as to how those GMOs can get put into products, so on and so forth. But there's other issues as well. So questions about cross-contamination. If I'm talking about crops, can those crops then pass those genes on to a neighboring crop field? Lots of things that people have concerns over, and rightfully so. This is something that, we're not necessarily used to doing, and so what are the repercussions? We should be aware of addressing those. That being said, I think a lot of the controversy around them, the scientific community has done a good job taking care of the concerns, whether or not they've been particularly good about communicating those safeguards and things that they are doing to ensure that these issues aren't issues. Maybe that's not necessarily getting across as well as they'd like, but once again, if we don't question what things are being done, then we can never ensure that everybody is being taken care of and done so safely.
Hello, I'm Hank Green, and this is SciShow. So we made a video about this once before, but some of the studies we cited turned out to be bunk, and in general, I think we played our cards too close to our chest when it comes to how we really feel about genetic engineering here at SciShow. So, why are GMOs bad? They're not. They just aren't, not intrinsically, and certainly not for your health. We've been eating them for decades with no ill effects, which makes sense because a genetically modified organism is simply an organism like any other organism that produces tens of thousands of proteins, but one or two of them were proteins that were chosen specifically by us humans. Genetic engineering is necessary for the continued success of the human experiment here on planet Earth, just like the advent of nitrogen fixing allowed for more fertile fields that saved millions from starvation. The fruits of genetic engineering, sometimes literally, will help us face the significant challenges of a world with more and more people and a climate that is less and less stable. Of course, just like nitrogen fixing allowed Germany to build bigger bombs, genetic engineering is a tool that can be used for good or for evil. So yes, it must be studied and controlled and understood, but that understanding has to start with, like, us. Right now. If you live in the United States, you almost certainly eat genetically modified organisms, or GMOs. Thus far, it's just plants, though pretty much every kind of meat on the market was likely fed with GM corn at some point. And it won't be long before the animals themselves are genetically modified. In 2012, the FDA reviewed a new kind of Atlantic salmon engineered to have higher levels of growth hormone using the genes of Pacific salmon and an eel-like fish called the ocean pout. They concluded that the engineered fish was safe and opened up the discussion for public comment, but still haven't announced a final decision. GMOs are everywhere in the US. Pretty much literally, 95% of sugar beets, 88% of corn, 94% of soybeans grown in the US contain traits like being insect resistant or herbicide resistant that were engineered into them. And some crops are genetically modified simply for human benefit. Around 500,000 children go blind every year because of a vitamin A deficiency. So a strain of rice has been developed that, unlike normal rice, contains enough vitamin A to keep children healthy. Or healthier, anyway. Now, the term genetically modified organism is actually somewhat of a misnomer. I mean, people have been genetically modifying organisms since the invention of agriculture. Every plant and animal species has natural genetic variability, and for thousands of years, we've harnessed this variability by practicing artificial selection. We cultivate and breed organisms to emphasize their most desirable traits. Cows that produce more milk and squash plants that survive drought. Brassica oleracea, also known as wild cabbage, has been bred so intensively that it is the wild ancestor of half a dozen different garden staples, including broccoli, cabbage, cauliflower, Brussels sprouts, kohlrabi, and kale. Corn originally looked like this. Over years of selective breeding, we have turned it into a massive, crazy, giant mutant version of itself that we happily throw on the grill without thinking of the centuries of breeding necessary to turn a grass seed into a sweet and starchy masterpiece. But when we talk about GMOs today, we're actually talking about genetically engineered organisms, or transgenic organisms. We're talking about genes from one species being extracted and then fused into the genome of a different species. This is called transgenesis, and though not all GMO food is created this way, transgenic crops are by far the most common kind of genetically engineered organisms you come across. But here's the thing, engineered organisms aren't new either. We've been tinkering with food in laboratories for nearly 100 years. In the 1920s, scientists realized that they could cause mutations in plants, thereby creating more genetic diversity and possibly more desirable traits by exposing them to X-rays and gamma rays and various chemicals. Through the 1970s, these methods of mutation breeding were quite popular and completely unregulated and largely ignored by the public. Republic. Thousands of cultivars produced this way are currently on the market. It's a kind of brute force hack. Just mess the genes up, plant the seeds, and see what happens, and then breed the cool new traits back into various strains of crop. Then, in 1983, scientists pioneered a new tactic where they successfully took a gene from an antibiotic-resistant bacterium and spliced it into the DNA of a tobacco plant. Now, of course, antibiotic-resistant tobacco doesn't have any real purpose, but it did prove that single gene transfer was possible. The new practice of transgenics was born. Now, the GM industry wasn't really able to take hold until 1994 when the USDA approved something called the Flavor Saver tomato, a fruit invented by a California biotech company that was altered so that it took longer to ripen, giving it a longer shelf life. It was the first genetically engineered crop sold to consumers. The Flavor Saver, though, didn't last very long, partly because people didn't like the taste, and partly because others, mainly in Europe, were suspicious of its genetic alterations. The Flavor Saver and its non-ideal flavor touched off a debate that continues to rage. Today, most GMOs aren't found in your produce section like the Flavor Saver was. Instead, more than 90% of commercially grown GM foods are commodity crops, staples like feed corn and soybeans, which have been modified to resist herbicides or insects. These crops are used to make the ingredients in lots of the processed foods we eat, or are used as fodder for animals that we later enjoy consuming the flesh of. Probably the most well-known of these transgenic crops are the so-called Roundup-ready crops, foods like soybeans, corn, sugar beets, cotton, alfalfa, and canola that are engineered to resist the active ingredient in the herbicide Roundup. These crops provide us with some, you might say, digestible examples of how transgenic foods are engineered, why they're made the way they are, what they do, as well as what they don't do. Let's start with why they were made in the first place. The active ingredient in the herbicide Roundup is glyphosate, a chemical that inhibits an enzyme plants use to synthesize amino acids. By blocking this enzyme, Roundup stops plants from making what they need to grow and metabolize food, thereby killing them. And it pretty much takes no prisoners. So much so that it can be hard to use around plants that you don't want to kill, like your crops. So in the early 1990s, the company that makes Roundup, Monsanto, decided to develop crops that were resistant to glyphosate, so farmers could spray the herbicide over their whole crop, but only kill the weeds. See, there are these microorganisms that produce an enzyme that is unaffected by glyphosate. All Monsanto had to do was transfer those bacteria genes to food plants, and farmers could use Roundup to protect their crops without killing them. So they extracted small pieces of bacterial DNA that were responsible for making the enzyme and set about introducing them into plants. But how do you get the genes of a bacterium into the nucleus of a plant cell? On the tree of life, plants and bacteria are not even on the same branch. Well, it turns out that there are a couple of pretty interesting ways. The first involves gene guns. Yeah, you heard me. Gene guns. Gene guns do pretty much what they sound like, literally and kind of haphazardly blasting DNA into plant cells. Most commonly used to engineer corn and rice species, they start with tiny particles of gold that are coated with hundreds of copies of a desired donor gene called a transgene. Cells from the plant that's going to receive the new genes are put into a vacuum chamber and then fire away. The gene-covered gold particles are shot at the cells using high-pressure gas. Once inside the nucleus of a plant cell, the gold dissolves, and the scientists cross their fingers and hope that the DNA is taken up by the chromosomes in the nucleus, which it sometimes is. Once the transgenes have been incorporated into the plant's DNA, it can then be bred into offspring plants. It's not exactly elegant, but it's a heck of a lot more subtle than just bombarding the seed with radiation and hoping for the best. Another more recent and more effective way to create transgenic organisms involves using a soil-dwelling bacterium called agrobacterium. This is a plant parasite and a natural genetic engineer. It has an extra and quite special piece of DNA called a plasmid that can move outside the bacterium and implant itself into a plant cell. In nature, the agrobacterium uses this little trick to recode plant cells to grow food for it, but in the lab, engineers can use the plasmid as a kind of carrier for fancy transgenes, using it to infuse plant cells with new genetic material. So, whether you've used the agrobacterium or the gene guns, you now have a new engineered crop plant. But you can't just put that thing into the ground. You have to introduce this new genetic material into existing traditional strains of the crop. This last step, called back crossbreeding, involves repeatedly crossing the new transgenic plant with breeding stock over and over again until you wind up with a new transgenic crop. At the end of the process, Monsanto had a patented plant that could be sprayed with glyphosate and survive. Previously, plants would have to be seeded far enough apart that machines could till away competing weeds, increasing soil loss and costs to the farmer, not to mention fuel consumption. Plus, Monsanto gets a whole new massive customer base for glyphosate. It's a long process. The whole thing can take as long as 15 years, but that's how just about all genetic engineering is done to your food, whether scientists are putting a bacterium's antibiotic resistance into a tobacco plant or an eel's growth pattern into a salmon. Of course, then there's the process of getting the crop or animal approved for use, which can also take quite a number of years. At the moment, it's extremely expensive, though there are some technologies on the horizon that might make it cheaper. The fact that it's so expensive and yet still economically actually worth doing indicates how extremely useful GM crops can be. It also means that the companies that produce them closely guard and restrict the patents and sale and growth and even research done on the crops. One of the reasons engineered foods are attacked so viciously is not because of the scientific consequences of their existence, but the economic and cultural consequences of placing so much power over our food supply into the hands of very few, very large companies. The GMO debate has become something of a surrogate for a much larger debate about economics that frankly is out of our league. There are some scientific concerns about genetically modified food. How does inserting a single gene, for example, rather than swapping out huge chunks of genetic material affect the genome at large? We used to think not at all, but it turns out that the genome is more complicated than that. Additionally, many farmers save non-patented seed for next year's crop, something you can't do with patented GM crop seed. But if your public domain seed was unintentionally fertilized by a patented strain, you might find that suddenly the seed you saved from last year's harvest to plant next year has genes owned by someone else, someone who is, it turns out, suing you. And if your livelihood depends on selling certified organic crops or selling into markets where GMOs are prohibited, the consequences can be even more dire. And of course, the traits we're engineering into crops might have potential ecological effects. Like if we're engineering in insect resistance, we want to make sure that we're not harming the insects that we do like bees and butterflies. But after having been consumed in hundreds of millions of meals by me and probably by you, and having been studied for decades, there has been zero indication that genetically modified food poses a danger to human health. That has not stopped an extremely vocal opposition from funding poorly designed studies and publishing misleading papers. We here at SciShow even reported on a study indicating that GMOs caused an increase in cancer in rats. This study, led by a guy who was not coincidentally publishing a book on the topic that same week, was published in a peer-reviewed journal and was initially taken at face value. But cherry-picked data, a lack of dose response, small sample groups, and a strain of rat that has an 80% chance of developing cancer in its lifespan eventually combined to completely discredit the study. Of course, as with any new technology, it can have unintended consequences. It can be controlled and monopolized and even weaponized, so there's plenty of reason to keep an eye on the companies making these advances. But when considering the number of hungry people on the planet, we have an obligation to explore every possible avenue to increase crop yields and decrease the amount of herbicide, pesticide, energy, and water needed to produce a crop. Traditional and advanced breeding methods need to be a part of that, and so does genetic engineering. Thanks for watching this episode of SciShow, and thank you to the people who pushed me to write up a more complete and accurate version of this episode. If you want to continue getting smarter with us, you can go to youtube.com/scishow and subscribe.